Hi hires, so we are looking at sustainability and interdependence unit still, and in particular Area 2, plant and animal breeding and heredity. So we're taking into account some stuff from Nat 5 and some other stuff that we've already looked at in higher and bringing it all together. So as I say, we've got some stuff that we just need to pull back up from Nat 5, and we've already looked at quite a bit to do with heredity, but more of its impact on traits because we've looked at selection. So just to remind you, every inherited characteristic you get through your genes. The variation can be either discrete or continuous, that's Nat 5. So discrete variation is where you put the characteristic that you have, the phenotypes that you see, into distinct categories. Okay, it will almost always be controlled by a variation at one gene locus. Okay, and that is therefore monogenic. Okay, so here's an example of discrete. There are only certain blood groups that you can have, and that's what we see. So continuous variation is where it's over a range of values. You should know that from, again, that's Nat 5. And that's going to be more complicated to try and control because it's going to be lots of genes that are involved. And then at each gene, you then have lots of alleles. So it's polygenic this time. And because of this, it's going to be on a curve. So this is the kind of curve that we've seen a lot. In fact, I pulled it out from the PowerPoints from the first unit, from evolution. So we've got increasing value on the bottom here, just increasing up this way. And then we've got the increasing number of individuals going up this way. And so this mean could be, you can shift it up or down, and this might be a really narrow bell-shaped curve in here, or it could be a much broader bell-shaped curve. You can do any of those. Okay. A bit of revision from your first unit. Okay, so selective pressure could be used to move the distribution curve. So we could either use disruptive pressure, so that means that we're actually selecting for either extreme of the mean, or of the curve, and that means that you can actually end up with two separate means. I'm using the word mean quite a lot there, apologise. Directional pressure means that we shift the mean. So in this case, this would be directional pressure for an increasing value. Okay, but you could have also directional pressure which was shifting it back the way. And then we have stabilizing pressure, where we get rid of the outside edges of this. So we're really very much selecting for the central point here, and that's meaning that we're getting this kind of moving in on either side. Okay, this selective pressure, we can actually push that, and that's the idea behind plant and animal breeding, is to choose what we want. So we have domesticated animals and plants thousands of years, and what we've done is we have constantly picked the particular parents who show the particular characteristics that we want and breeding for them. And what that means we end up with is artificial selection. So this is either artificial selection or selective breeding is our new term. Okay, and then you end up with specific characteristics, and because it's selective breeding, we then call them breeds if it's animals, and if it's plants, we call them cultivars because we have cultivated them. Now, if we want food production, because remember this is all about really food production ultimately, then we need to have really high yield cultivars, and we need high yield animal breeds as well. So current food production depends really on making sure that we have the right breed and the right cultivar. So we look at, first of all, heritable characteristics that you could actually select for, and you can do just about anything. I mean, here's a list, but I'm sure you could come up with more. So this is one of the most common examples to give you of artificial selection. We have a wild mustard, so how everything started off, and then we've got selection pressures that have been different. This is us, that have created totally different vegetables. So if there's been selection for the flower buds, we've ended up with broccoli. The terminal buds, so the one at the end basically, gave us cabbage. We've got root selection gives us turnips, stem gave us kohlrabi, flower buds again, but in a slightly different way than the broccoli, gives us cauliflower, and the leaves gave us kale. Okay, so these are all related vegetables, but now they are totally different in terms of how they present. This is just a kind of, I think, reasonably clear example of how we've done quite drastic selective breeding this is obviously in cattle and in chickens. So discrete variation you can actually monitor quite easily, and you can use crosses to do this, and you're still expected to be able to do monohybrid crosses. So if you think that this is a bit dodgy and you're not entirely sure of it, go back to the Nat5. It's multicellular key area 4. Okay, so there's videos on that one, and if you need any extra help in it, then just let me know and I can put some more things up. So higher, you need to have just a few more definitions that are a little bit clearer. Some of them are the same. So Nat5, we talked about test crosses or back cross. So what you do there is you have an unknown parent, but you know that it has the dominant phenotype. Now what that means is that you have something that is either that or that, but you don't know which, because as soon as you had one of these, they look exactly the same. So what you do is you cross it with the homozygous recessive, and what you get tells you. Basically, if it's this one, you'll get a 50-50 dominant recessive in the phenotype, and if it's this one, you still get 100% of the dominant, and that tells you what the parent was. Okay, so that's still as Nat5, but obviously higher question context might be a little bit trickier. True breeding, still same as Nat5, so what that means is that you have a homozygous genotype, because what that means is that it will only ever pass on the same thing. So it's either this or this. Now this is definitely higher, we haven't talked about this or used this term. Inbreeding means that parents are drawn from closely related. Okay, outbreeding means that the parents that you're picking are unrelated. Now these are really important when it comes to the health genetically of the group that you're looking at, so we will talk about these a little bit later on. And then crossbreeding means that they are from different breeds of a species, so you're actually picking from quite not just outbreeding but drastically different. So the whole point of the breeding is to try and get consistently the characteristic you want. Now that is hard, because what you're also trying to get rid of to make this happen is you're trying to get rid of heterozygosity. Now if it's a monohybrid, this is really, and it's only got 2 forms of a gene, what we're really saying is get rid of these guys, the heterozygotes, and end up with, well, let's say it was the dominant that you wanted. You only want this in the breeding population. You don't want, well, you certainly don't want this, because that's not what you wanted at all, but you don't want this. Because if you have this in here, then every so often you're going to get these alleles that are recessive, so they don't show up all the time, but then they do occasionally. So this is not what you want this. Now the problem is this is not reality. The reality is most of the things you're looking at are polygenic with more than one different potential alleles. So you know, this one is just 2 alleles, big A and little a, but it could be hundreds. Not at one loci, but if you take in all of the polygenics that are there. So the more heterozygosity you have, especially in polygenics, the more middling, intermediate phenotypes you get. So say you were looking at petal colour and you wanted red, and it runs from red to white, but it's polygenic. You have everything in between going from red to white. And so what you're trying to do is to get rid of all of the alleles which support white and leave you with just the alleles that support red. That's just a picture of some ladybirds.